Hello and welcome to this BioProcess International Ask the Expert webcast. As I mentioned, I'm your host, Jordan Stillman, and I'm project coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. Please enter your questions at any time. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you so much for joining us today. It is now my pleasure to introduce our speaker, Bernardo Cordovez from Halo Labs.
Thank you so much, Jordan, and everyone. Thank you so much for joining us in this webinar today. My team and I were so excited to introduce the AURAT to the world. It's an amazing product and probably the only subvisible particle tool that you're going to need in order to determine the stability and the purity of your biopharmaceuticals. The main objective of this product is for you to find your particle problems before they happen, because right now the current instrumentation does not allow you to do that, and we're trying to revise that. So the mission of this product line is for you to know your particles, and what we mean by that is to know 100% of what's in your sample is related to particles, what they look like, and most importantly now, what they are. And one reason why that is very important is that we took a poll with our current and prospective customers. And the number one question in protein therapeutic particle analysis that comes up is that particle a protein or not? And the reason why that question is very important is because it is the point in which formulators make the decision in understanding whether they have a stability issue or if something else is going on with the formulation. But that's a little bit challenging with the current tools that you have available today. So in this slide, what you see is 3 sets of images. And actually, 2 of these sets of images are actually proteinaceous. And this is a high-impact paper published by the Japanese Pharmacopoeia. And I'm going to give you a few seconds to guess which 2 sets of these 3 images are actually protein. And if you guess these 2, you have much better eyesight than I do. As you can see, using morphology, shape, and intensity is simply not enough for one to be able to distinguish between proteins and, for example, plastic particles or degraded polysorbate, which are very common in bioformulations. And this is what the imagers in a flow imager would look like. So clearly the flow imagers don't work. And so what is your situation at the opposite end of the spectrum? Well, that's spectroscopy. And this figure that you see on the left of this slide is actually a very good result. Even though it's a little bit complicated, it's a series of overlaid spectra. And for the untrained eye, it doesn't mean anything. But wait, actually, it's a really good result that took probably several hours to acquire. And there's the 1550 wave number peak, which is indicative of an Mi2 bond. So after shooting a laser and rastering and finding that one spot and cleaning up that signal, you get a little bump that indicates that there might be a protein there. So as you might imagine, that is a very laborious, it requires a ton of expertise, and it's quite an intense process that only very few people can do. And it's so low throughput that it takes a tremendous amount of time, a tremendous amount of training, and therefore spectroscopy tends to be relegated only at the tail end, in forensic once you've failed. So you can only do proper ID once things have failed, and that's simply unacceptable. And that's why we built this product, because we want you to find your problems as related to particles before they happened. And we created this amazing technique called the fluorescence membrane microscopy, which powers the AURAT instrument. When compared to other common identification technologies, we basically, we're far superior in every regard. So we're true high throughput. So we measure 96 samples in under an hour. If you wanted to measure, for example, with Raman, let's say every sample has 1,000 particles, every one of those measurements would take you 10 minutes, right? So to do that for a full high throughput screen, it's simply impossible. It would take you months to years. Our signal to noise is very high because we have a high refractive index measurement basis. We measure 100% of your sample. Our instrument is very easy to use. It's essentially a green button solution, but with a lot of software and great analysis underneath that allows you to do a very deep dive. One of our main value props is that we enable low volume subvisible particle measurement. So you can measure samples as low as 5 microliters in volume quantitatively, and now we can ID particles. So we're essentially superior to FTIR, Raman, and flow imaging in pretty much every regard. And the basic values of the AURA is that we can identify particles and distinguish them between protein aggregates and non. The system enables 100% sampling efficiency. It's quantitative down to 5 microliters of minimum volume. And to put that in perspective, something like a light obscurator can take over a mil, right? So literally 200 times less than the typical compendial method. We have a very large dynamic range from a micron to 5 millimeters. It's compatible with robots. It's high throughput. It comes in a 96-volt format. And it enables high refractive index contrast measurements, which is key because particles in a bioformulation are inherently very similar to the aqueous suspension that they reside in, and therefore it's very difficult to resolve them. Plus, we have a second channel that enables different kinds of identification. And this product comes essentially, it's equipped by 3 components. The main workhorse, of course, is the AURA instrument, which conducts a high throughput particle detection in bright field and in fluorescence. We are launching a new set of plates that support labeled fluorescent workflows, as I'll describe in more detail. And we have amazing acquisition and also intuitive and flexible analysis software that allow you to understand your sample from a single particle to high-level insights, as I'll show in the next few slides. So let's just start with sizing, counting, and imaging particles and how we're better than everything else that is out there just in those components, and then I'll move into particle identification in the second half of the presentation. So the AURA is also powered by background and membrane imaging, which is a proprietary technique that allows us to measure particles from 1 micrometer to 5 millimeters in size by imaging onto a membrane. And it's inspired by USP 788, which is a compendial method for measuring subvisible particles that are particles from 1 microns to 100 microns. And here's basically how it works. First, you take one of our plates and you load it in the system, and then you take an image. This is called the background, right? So membranes have topography, and that is captured in this backgrounding step. Then you literally take that plate, you bring it onto a proprietary vacuum manifold. You pipette your sample. Then you suction the sample through this filter plate. Again, you can't see them, but it's a polycarbonate plate with 400 nanometer pores. So we're essentially filtering the sample through, and we're only going to have the filtrates, right? The particles larger than the pores are going to remain behind, and then we're going to image those. So as you can see now, we have an image which is a combination of the membrane background as well as the particles. But as you may remember, we already took an image of the membrane to begin with. So then using really powerful proprietary software, we aligned the images and subtract the background, making it possible for us to resolve particles that without this key step would be impossible to resolve. So now we can measure particles down to a single micrometer with high refractive index contrast, which is something that is incredibly important, again, because biopharmaceutical particles have optical properties that are very similar to the liquid. They're almost essentially invisible. And in addition, in a method compliant with USP 788, which is now updated to accept image analysis on membranes as part of a compendial subvisible particle methods. So one of the great features of this system, again, is that it gives you all the details that you need from the single particle level. As you can see, this is a small particle, happens to have 57 microns in equivalent circular diameter. There's parameters that's related to its shape, to its 3D morphology. We have a secondary light source that allows you to understand roughly how tall your particles are. So that's at the single particle level. Then let's look one level up. And this is typically where the state-of-the-art instruments, that's kind of where they finish. At most, they can spit out a particle size distribution, as you see in the top. So for a given sample that you pipette on a single well, you're going to, this system is going to tell you how many small particles you have and how many big ones you have. But in addition, we have amazing data visualization built in. And then if you do a whole plate or an experiment, it has very intelligent software that combines all of this data under one roof and allows you to extract high-level insights, so CVs, replicates, etc., so that you understand what is working, what is not, what is stable, and what is not stable. All the while in high refractive index measurements, which again, if you look at flow imagers or light obscurators, the particles look identical to the surrounding media, but by draining essentially the surrounding liquid, we can enable high contrast measurements. But we've even improved the existing method. And now background and membrane imaging in AURA has 100% sampling efficiency and a very, very powerful software that allows you to analyze your particles in your samples and with much higher fidelity. So 100% sampling efficiency matters. So in the case of our old original system, the Horizon, we only sampled about 63% of the membrane. That meant that we needed scaling factors to sample the whole sample. The AURA images the entire well, so that's 100% sampling efficiency. And because all of the particles are in focus, that means that 100% of the particles that are filtered are going to be in the image plane and properly imaged. If you compare that to a flow imager, only about 20% of the particles are in focus because your channel heights for those types of systems or even light obscurators are, say, 300 microns, and only the focal spot is only a slither of that. In the case of flow imagers, about 60 microns. There's no data acquisition knobs. You don't have to set flow rates. You don't have to set exposures. And it has a dramatically large sizing and counts dynamic range. So in this image right here, we can see that for 1.6 micrometer beads, it's linear above 3 million particles per mil. So we can measure not only small particles, but the linearity is also quite high. And most importantly, for earlier in formulation, when you're dealing with difficult to make samples, or you don't have many samples, or even tricky samples, for example, like AAV formulations, you don't have a lot of precious sample to use all over the place. And what we're showing here is that these measurements are linear down to 5 microliters per well of sample, and with really strong linearity as well. So we've basically proven that it is quantitative and statistically relevant even at the smallest volumes. And that's just sizing and counting. But the reason why many of you are here might be, okay, so what about identification? And the AURA enables high-throughput protein ID via fluorescence labeling. And essentially, the black and white image that you saw before in BMI is now a colored image. In this case, the red particles are protein particles, and the blue particles are non-protein particles. But so we're going to, in the next slides, I'm going to do a deep dive to understand you how our fluorescence protein ID capability works, and even expand on that towards the end of the presentation. So the workflow is very similar to what I presented to you at the beginning of this presentation, right? So the first 3 steps are basically BMI. You take a picture of the membrane topography, you background the plate, then you apply and suction your sample, and then you measure it in bright field, right? So the particles are detected using the standard imaging source. So at this point, you're going to have what I showed before, which is like the single particle, the sample level information, even your experimental level information. But the system doesn't yet know if those particles are protein or not. At that point, we're going to take the plate, we're going to bring it back to our vacuum manifold, and we're going to apply 40 microliters of dye. In this case, it's going to be THIOFLAVINT, which is the most well-validated dye for conducting protein aggregate staining. We're going to suction it, and without having to wait pretty much any time at all, we bring it back to the system and we measure it in fluorescence. And the system is so amazing that it puts it all together for you, and it now is able to understand which of those particles were proteinaceous to begin with and which of those were not. So one experiment that I'm going to show you is the ability to distinguish the undistinguishable. So NIST, the U.S. National Institute of Standards and Technology, has made this protein aggregate mimic out of plastic. It's called ethylene tetrafluoroethylene, ETFE. And I don't know if you remember from earlier, but one of these particles is ETFE. It's just very difficult to know. So it's a place where morphology can tell you if it's a piece of plastic or if it's an actual protein. So let's use that as a case study. So in this experimental setup, we're going to do a whole plate, almost a whole plate setup, in which we're going to run 24 wells of rotated human IgG, so stress IgG particles. We're then going to use this plastic shards made by NIST, this mimic protein aggregate, in another 24 slides. And then in 24 wells, sorry. And then in columns 7, 8, and 9, in the next 24 wells, we're going to do a mixture of these two. And that's going to be a serial filtration of ETFE and IgG. So I'm first going to pipette ETFE, and then I'm going to pipette IgG over it. And therefore, you know that the total number of particles that you're going to have there are the sum of ETFE and IgG. So that's a controlled experiment, as well as the corresponding water controls. So let's play this out. Can we distinguish the IgG and the ETFE portion correctly, and even just the ETFE from the IgG? So again, this is a controlled experiment. We can, before we do any fluorescence, we can just look at what the particle counts for these different combinations of materials look like. So the ETFE, you can see that you're measured about 50,000 particles per mil, greater than 2 micrometers in size. For the IgG, it's on the order of 140,000 particles per mil. And for the serial filtration, for this first time we pipetted ETFE and then IgG over it, we got about 185,000 particles. So it's pretty much dead on that IgG plus ETFE, the serial filtration, shows you, corresponds to the single non-mixed samples, ETFE and IgG. So we can measure that. So that's great. We're in a good place to start our experiment. We're then going to take those that very same plate, and we're going to stain it with 5 millimolar thioflavin T. And again, thioflavin T is one of the best validated staining dyes, inspired by several decades worth of work in neurodegenerative disease research, including Alzheimer's and Parkinson's. And it tends to bind to misfolded beta sheets. So, and particularly the amyloid portions of protein aggregates, which pretty much every protein aggregate at the subvisible stage exhibits. So here's two images. The red particles are the saturated protein fluorescence images from saturated fluorescence images from protein. And you can see that on the left image, we basically just pipetted 40 microliters of 5 millimolar THD, and the particles saturated in fluorescence, while if we waited for 3 minutes, they also saturated. So the long story short is you don't need to sit around and wait. This system experiences some really interesting kinetics, that everything happens very, very quickly on the membrane. And if we actually look at these fluorescent images, our water control looks pitch black. Our human IgG rotated aggregates, they're showing blended images of fluorescence in red. At the bottom right, ETFE, you're looking at not a fluorescence, but actually a side angle scattering signature in blue. And in the bottom left, it's a mixture of these two. So you can see this ETFE particles as well as the IgG, ETFE in blue and IgG in red. So we can very quickly see what do these particles look like and separate the two of them just visibly. But now it's time to do some quantitative analysis. So this graph plots fluorescence intensity in the y-axis and size in the x-axis. And each one of those dots is a particle. And what this shows you is that for human IgG, you can see that at the bottom of the y-axis, where it goes essentially from, say, 10 to 250, and that's essentially the range of an 8-bit image, if you know a little bit of optics. So 255 means white and saturated, and 0 means black. So as particles get bigger, you can see that there's more binding sites, and typically fluorescence grows. So human IgG is staining, and we can quantitatively look at how much each one of these particles is staining. In the case of this protein mimic, again, it's not a protein, it's plastic. And you can see that every single one of these dots in the scatter plot has very low fluorescence. So it's basically not fluorescing almost at all. And there's very clear differences in the fluorescence signatures for these two particle types, even though by morphology, we couldn't even tell them apart. And if we actually look at a mixture of these two, you can see that it's a combination of particles that exhibit almost no fluorescence versus those which do. And essentially, ETFE, they are black. They are not showing any fluorescence at all, where the human IgG aggregates with these amyloid structures are binding to the THT and therefore fluorescing strongly. So we have a whole host of ways of not only looking at the images, but plotting them in the scatter plots for you to see single particles that stain or do not stain. But we can draw some amazing conclusions using thresholding analysis and Booleans. And it's very simple. In this case, you can see I have a vertical dash line and a horizontal dash line. And that splits up this data set into 4 different quadrants. And Q2, and as for this frame of reference, is the top right quadrant. And we're going to interrogate how many of the particles are in this top right quadrant. That means particles that are bigger than 5 micrometers in size and are exhibiting fluorescence above where these ETFE non-fluorescing particles are. Now let's take a look. In the case of ETFE, we measured about almost 16,000 particles per mil greater than 5 micrometers in size. But only 244 of those were above this threshold, meaning that essentially only less than 2% of the ETFE fluoresced. And it could have been even something else. In the case of the human IgG, however, for these 43,000 plus particles, almost 100% of the sample stained at 99.8%. While in the case of these serial mixtures, about 88% of the particles cross this threshold. And again, this can be done manually or with even built-in expressions, which come with the system that allow you to do this fluorescence-based separation. Now, this 88% number might look a little bit strange, but that's where we ran a controlled experiment. And as you might remember, the IgG plus ETFE serial filtration, I first filter the ETFE and then we filter the IgG over it. So we know exactly how much material is in there. When calculated by counts, the ETFE portion of this mixture is about 15.5%. So the non-protein portion of the mix is 15.5% for greater than 2 microns and 14% for the greater than 5 micron bin size. But if we actually do this calculation by fluorescence, we get exactly the same results, right? So if we wouldn't have known what the sample was beforehand, and we've only would have ran this in this fluorescence-based analysis, we would have ended up with the exact same results with an error. So it's an incredibly powerful method, and it's been verified and validated in several ways. There's more than one way to look at this data. For example, I was showcasing before fluorescence in the y-axis and size in the x as a way to create these scatter plots to gain greater insight of these individual particles. But if we now plot fluorescence versus side illumination intensity, which is essentially our proprietary dark field technology to look at 3D topography of particles. So if they stick out of plane, they're going to have a strong side scattering signature. You can see that the human IgG particles, they clearly grow in fluorescence from 0 to saturation, but they do not exhibit pretty much any side angle scattering. While in the case of this ETFE, this protein plastic mimic, they don't exhibit any fluorescence. Of course, they don't have any amyloid locations in which a dye can bind to, but they protrude out of plane. So what is the summary here? That these particles couldn't be any more different. And if you were looking at this with a flow imager, they would basically look identical. So we have several degrees in which we can produce high fidelity separation of even the most complicated particles that look identical. And it works for some of the most important controls. So polysorbate particles, they don't stain in the THT. Many of potentially your particles come stored in Corning cryovials. They tend to shed particles like crazy, but those do not stain, so they would look like a non-protein. And same thing for other particles, including stainless steel and other common particles in the bioprocess. So the controls hold. And in addition, there's even more fluorescence options available for you. So we have a second channel that can be used to identify lipids and protein monomers, et cetera, that we have some built-in preset settings, as well as giving you the ability to choose to pursue your own application there. So this image on the right is an example of a mixture of human IgG particles mixed with palmitic acid particles, and we're looking at a multi-channel fluorescence, and as well as a side scattering signature to see which of these particles actually have a 3D topography, which of these particles protrude out of plane. So we can start now doing a whole combinatorial separation and categorization of particles in which we can, or allows you to understand what the protein component element is, what the non-protein component element is, which of these particles are hydrophobic or lipophilic, and how many of those are actually protruding out of plane. And that's all very, very fast. So we have, the system comes in with built-in default channels as well as second channel options that adapt to your needs. And please feel to reach out so we can talk about that in more detail. But why Aura? Honestly, the true question should be, why not Aura? We enable high throughput particle analysis. So we can do in less than 2 hours what it takes the other instruments a few weeks. When it comes to particle identification, we can do in a few hours what it would take other instruments a few months or a year. And that is basically how it is. In terms of low sample consumption, we're quantitative down to 5 microliters as opposed to 500 microliters, which is typically what the other instruments require on the minimum side. We measure 100% of the particles. We enable specific high to low level insights, starting from a single particle all the way to an experiment level combined data insights, producing particle size distributions and counts analysis. It's fast and flexible. It comes with 21 CFR Part 11 software. The images have high fidelity because they're high contrast images because of the high refractive index measurements. And it's dramatically easy to use, and it can do a lot more, which we're basically just scratching the surface. So really appreciate you joining us today, and I'm now happy to take your questions.
Fantastic. Thanks, Bernardo. So we do have some questions, and let's start with, can the second channel be used for other protein dyes?
That's a good question, and the short answer is yes. And we have demonstrated the ability to work with monomer dyes as well as THT. One of the popular ones is Alexa, both the NHS ester and the Mali Emmet Alexa. One of the reasons why we can do it in the system and not in flow-based systems is because we drain the liquid, which is full with monomer, which would increase the background of any, for example, like a cytometer, right? You would just see the entire background glow. But in this case, we drain that if we only have the particle aggregates remaining. And so the short of it is yes, and we have some data doing this with the Alexa dyes.
Great, thanks. So are you limited to THT dye? What emission wavelengths could be measured with the instrument?
So no, we're not limited to THT dye. The THT dye is just the first channel, and we're happy to get to you with more details. We have a second channel available, which is either customizable to whatever you happen to have in mind, but the 3 default channels that we have so far are for BODIPI 630, TMA, DPH, and dye. Most of them target lipophilic particle fluorescence-based identification. But in addition to that, we have the flexibility to customize that further should that be important for you. And so please reach out and contact us for more information on that.
Great. And how much extra time does the fluorescence measurement add?
Only about 30 seconds per sample. So yeah, that does basically only 30 seconds per sample. And again, comparing that to the incumbent technologies like Raman, that is about several minutes per point, right? Here we analyze the entirety of a sample in fluorescence mode with just another 30 seconds of time.
All right, and then next, see what volume of dye is needed to sufficiently label the particles?
That's a good question. About 40 microliters of dye to fully immerse the well with dye material. So 40 microliters per well.
Fantastic. Okay, and forgive me if I do a mispronunciation here, but what role does SIMI, S-I-M-I, play now with fluorescence?
So I talked about SIMI a little bit in the presentation, the site illumination, which gives users insights on the 3D topography of the particles. So SIMI is not used in the fluorescence fashion, so we're not doing fluorescence via SIMI. However, as you may recall in the experiment that I presented of separating this protein particles versus these plastic particles, we used the 3D topography information to further confirm that they were in fact not proteins. So it's not a fluorescence-based approach, but it does give users additional morphological information that would be essentially impossible to obtain with flow imaging systems. So it's a great complementary tool to further characterize your particles.
Awesome. And I think we have time for one more question. So can any measured parameter be used to search in the expression engine?
Yes, and I touched on the expression engine briefly when we did the protein versus non-protein separation. And the short of it is yes, because all of the parameters are stored for all of the particles. Their size, their area, their SIMI intensity, their fluorescence intensity, their ferrite diameter, their circularity, everything is known. And we can use this expression engine to basically determine, hey, how many particles meet this specific criteria? And again, all of the parameters are stored for all of the particles in all of the wells. So yeah, it's an incredibly powerful tool that we have, and it exploits all of the information that is stored in the system, which is basically every single parameter acquired for these particles.
Fantastic. Thank you so much, Bernardo. And thank you to our audience.
Thank you.
Well.
I appreciate it.
No, no problem. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you will receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Have a wonderful day and goodbye.